I've divided L9 into three different mini presentations. The first mini presentation, L9a, will involve DNA structure, and we'll start with the central dogma of molecular biology and collinearity, and then we'll go to the structure of DNA and levels of organization. L9b will be replication, a view at the molecular level, and we'll talk about DNA replication in prokaryotes, DNA replication in eukaryotes, and then the ends of the chromosomes, telomeres, and telomerase. L9c, part 3, will be correcting mistakes and mutations. We'll discuss 3' to 5' exonuclease activity during replication, base excision repair, DNA glycosylases, nucleotide excision repair, double-strand breaks, and mutagenesis, and the Ames test. So L9a, central dogma of molecular biology, flow of biological information from DNA to RNA to protein. And here we have our double-stranded DNA, and this section right here, the double-stranded DNA, contains the hexokinase gene. We transcribe one of these DNA strands to get a complementary RNA, which contains the coding sequence for hexokinase, the protein. Translation of the mRNA to an unfolded polypeptide gives us the amino acid sequence of hexokinase. And then upon folding, we make the catalytically active hexokinase. One of the terminologies we're using here is something called collinearity. And collinearity means that we have a coding sequence on DNA, and the coding sequence on DNA corresponds to the coding sequence on an mRNA, which corresponds to the amino acid sequence that we get upon translation. Here's our anti-parallel double helical DNA. We're not showing it as a helical, we're showing it in a linear form. And the 5' to 3' chain has the coding sequence. So it's CGT, GGA, TAC, etc. And we make an mRNA, but when we transcribe the mRNA, we actually transcribe the complementary strand, which is running in the 3' to 5' direction, in order to make the 5' to 3' mRNA. And each one of these triplets is a codon, and during protein synthesis, we'll study more about codons. So this is a phenomenon called collinearity, which is the sequence from DNA, gives us a sequence of RNA, which gives us a sequence of protein. Here we have Watson-Crick classical base pairing, and this is showing A base pairing with T, and this base pairing is determined by hydrogen bonding. Here we have the carbonyl group from carbon 4, and its hydrogen bonding with the hydrogen of the nitrogen from carbon 6 from the amino group from carbon 6. And now we have the ring nitrogen number 1, which doesn't have a hydrogen. It has an electron pair, and it's hydrogen bonding with the hydrogen that's part of ring nitrogen 3. So here's ring nitrogen 1 with an electron pair. Here is a hydrogen. There's another ring nitrogen with a hydrogen. And then we have the GC base pairing. So here's a hydrogen bond. Nitrogen 1 has a hydrogen on G. Nitrogen 3 in C doesn't have a hydrogen. Here's electronegative, electropositive, electronegative. So this is a hydrogen bond. And then we have an amino group at carbon 2 of the G. And the hydrogen of that amino group is hydrogen bonding with the carbonyl group of carbon 2. So a GC base pair contains 3 hydrogen bonds. An AT base pair contains 2 hydrogen bonds, so it should be clear from that. If we have a DNA strand that has a predominance of G and C's, which would be complementary to other G's and C's running in the other direction, then we will have stronger interactions in that duplex, in that double-stranded DNA, than we'll have in a long strand of double-stranded DNA that has predominantly A and T, because we have a summation of 3 hydrogen bonds over many, many GCs versus 2 hydrogen bonds over many, many ATs. What is really different about cDNA? So we're going to talk about 3 different DNAs. We're going to talk about got B DNA, A DNA, and Z DNA. And first, this is our double helical DNA, and B DNA is the structure that was described by Watson and Crick and that they received the Nobel Prize for. Again, this is an anti-parallel structure where the phosphodiester backbones, this is the phosphodiester ribose backbone, this is the phosphodiester backbone. Just arbitrarily, I say this is going from 5' to the 3' position, and this is going from the 5' to the 3' position. So these two strands run anti-parallel. Here is our base pairing and the hydrogen bonding. So this has to be a GC because we see 3 hydrogen bonds. This is an AT. Here's our 2 hydrogen bonds. These are arranged like the rungs of a ladder, so they're pretty much straight across, and they're pretty much in the center of the double helix. We have what's called a minor groove between the phosphodiester backbone, and we have a major groove between the phosphodiester backbones. And notice there's more space in the major groove than there is in the minor groove, and often proteins that recognize DNA sequences that are going to perform certain functions like restriction endonucleases or transcription factors, promoter proteins, etc. They usually bind in the major groove because they have much more space, many more nucleotides to recognize relative to sequence. So there's more space in here. So anti-parallel, 10 bases per turn. So if we go all the way around a turn, there'll be 10 bases in right-handed helix. So this is a right-handed helix. And then we go to aDNA. It's also a right-handed helix. How do I know? Because these strands are running in the same direction. They're turning in the same direction. So this is a right-handed helix. This is a right-handed helix. aDNA is a little more compressed than bDNA. Notice there's less difference between major grooves and minor grooves. They're closer together. Here, the strands are far apart. Here, they're closer together. These are almost equal, and this is a little bit compressed since we have 11 bases per turn rather than 10 bases per turn. And aDNA is often found when we have a DNA segment base pairing with an RNA segment, or an RNA segment base pairing with another RNA segment. So rather than seeing a B structure, we see something that's more like an A structure. And then we have zDNA, and notice the zDNA, if we look at phosphodiester backbone, it's running in the opposite direction. It's slanted towards the opposite direction, and that means this is a left-handed helix. It's called Z, and Z is a shortcut for zigzag because the phosphodiester backbone, if we look down this backbone, these are pretty much running in a line, and these are pretty much running in a line. And with the zDNA, the backbone zigzags around, so it's zigzagging here and around here, zigzagging back and forth, zigzags over to here, back over to there, and zigzags over to here, and zigzags over to here. So the Z stands for zigzag, and zigzags are often found in DNA sequences that have a high predominance of alternating purines and pyrimidines. For instance, GC, 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 AT, AT, GC, GC. They may give you a left-handed structure, a zDNA that is being flanked by 2 bDNA regions. So we can have a long piece of DNA that has both bDNA in it and zDNA in it, because many regulatory regions often have repeating, alternating pyrimidines and purines. Just believe that maybe these are found in regulatory regions that bind transcription factors, etc., but that is yet to be proven. DNA, genes, and chromosome contents of some genomes. So E. coli, very simple, tiny bacteria, has 4,600,000 base pair, 1 circular chromosome, 4,435 genes approximately. Saccharomyces cerevisiae, 12 million. When I say the 12 million, I'm also converted into scientific notation because as we get to the end of this list, we're going to see some big numbers. So this is 4.6 times 10 to the 6th. This is 1.2 times 10 to the 7th base pairs, 16 chromosomes, a little more than 5,800 genes, 5,860 genes. C. elegans, 9.0 times 10 to the 7th base pairs, 12 chromosomes, 23,000 genes. So a huge difference between the yeast, and now we're getting to a more complex eukaryote. So 90 million base pairs, 12 chromosomes, 23,000 genes. Arabidopsis, which is a plant, 1.2 times 10 to the, this is to the 7th, this must be to the 8th base pairs. 10 chromosomes, 33,000 genes. Drosophila, your common fruit fly, 1.2 times 10 to the 8th base pairs, 18 chromosomes, 20,000 genes. Rice, 4.8 times 10 to the 8th base pairs, 24 chromosomes, 57,000 different genes. These are different coding sequences for different gene products. Mouse, 2.6 times 10 to the 9th base pairs, and for both mouse and Homo sapiens, this is the haploid number of base pairs. So if it has 40 chromosomes, this is the number of base pairs in 20 chromosomes, 27,000. And humans, 3.0 times 10 to the 9th base pairs in a haploid cell. That would be 23 chromosomes and 46 chromosomes. In a diploid cell, you'd have 6.0 times 10 to the 9th base pairs. 29,000 genes. I should mention, after the Genome Project, this number has been revised downward, and it's probably more like 22 to 23,000 genes. So of all this DNA we have in the haploid 3.0 times 10 to the 9th base pairs, only 1.5% of it codes for protein, is actually coding sequences for protein. And 28 to 29% of the DNA are actually intervening sequences and non-coding sections of those genes. And then we have miscellaneous sequences, and a lot of these are repeating sequences. They're repeating hundreds to thousands to tens of thousands times, very short sequences. And then we have retrovirus-like and transposon-like that make up about 45% of our genome. These repeating sequences make up about 25% of our genome. And the genes make up about 30%, but only 1.5% are composed of sequences that actually code for amino acid sequences. So that's interesting. This is an E. coli that's been lysed, and this is the DNA of that one circular chromosome in one E. coli that's streaming out of the cell. So we've disrupted the cell, and now it's released its DNA. And notice how much DNA you have in relationship to the size of the E. coli. And that means the DNA must be highly packaged and organized within this E. coli to make the one simple circular chromosome of E. coli. These round things like this, those are actually plasmids. You've probably heard of plasmids, which are small circular pieces of DNA that contain, for instance, antibiotic resistance. So these are called extrachromosomal pieces of DNA. They're not incorporated into this large E. coli chromosome. What we describe next will have to do with eukaryotes and how we package the DNA. And so here is our double-stranded DNA. Most of this, remember, is the beta DNA. And it wraps around protein complexes that are called histone proteins. And each one of these that has the DNA wrapped around it, this protein complex, is called a nucleosome. And the nucleosomes assemble into 30 nanometer fibers. So here's a 30 nanometer fiber, and we'll see some higher order structure in this later. And then these 30 nanometer fibers organize into what are called loops. So here's the loop, and then the loops organize. So this is a loop of a 30 nanometer, this is a loop of a 30 nanometer, this is a loop, this is a loop. They organize into 300 nanometer fibers, and each one of these that we see, these loops, organize, 6 loops organize in what's called a rosette, and they organize around a set of proteins that are involved in what's called the nuclear scaffold. So here's a rosette, here's another rosette, here's another rosette, and now we're starting to look like the wiring that's on a telephone. So eventually we organize into metaphase chromosomes. So this is the interaction of two chromatids. This would be a chromosome over here, and this would be a chromosome over here, and this would be happening during, say, mitosis. This could be happening during meiosis. So here's the histone core of a nucleosome with DNA wrapping around about 1.7 times for each nucleosome. And then we have a linker region between the nucleosomes, and we're going to find that we have one particular histone protein that's involved with this linker that helps attach these nucleosomes together. So types and properties of histones, we have histone H1, H2A, H2B, H3, and H4. And this shows you the size of the different histones. So H1 is the largest. These histones are positively charged because they contain large percentage of lysine, which is a basic amino acid, and arginine, which is a basic amino acid. And at pH 7, basic amino acids, the residues have positive charge because they haven't lost their protons yet. Histone H1 has 29% lysine, almost 30% lysine, 11 arg. H2A, almost 11% lysine, 20% arg, 16, 16, 19, 13, 10, 14. So notice H2A, H2B, H3, H4, almost the same size, and H1 is much larger. We're going to find H1 has a different function than H2A, H2B, H3, and H4. So here's H1 as we wrap our DNA around the histone. H1 tends to attach or to affix itself to the nucleosome and keeps the DNA in place. The octamer contains 2 H2As, 2 H2Bs, 2 H3s, 2 H4s, 2, 2, 2, and 2 makes 8. Remember, they're about the same size. They fit together. And then we have the H1. The H1 is called the linker histone. It links these DNA strands to each other, and it's also involved in the organization of histones interacting with other histones. So each one of these, this is a nucleosome, that's a nucleosome, this is a nucleosome, this is a nucleosome. So these are called beads on a string. And this represents supercoiling. So this is like a 30 nanometer fiber, and this is of course a phone card. So phone cards are great for showing supercoiling. So here we have coiling here, coiling within this, and then we have coiling amongst the coils. So it's coiling of coiling of coiling, and that phenomenon is called supercoiling. And this would be like one of the petals of a rosette. If we put 6 of these together, and this is the nuclear scaffold, then we would have a rosette that's part of the 300 nanometer fiber. So this shows the organization of nucleosomes that are going into the 30 nanometer fiber. And here we have 2 nucleosomes that are assembling, and then this is a 30 nanometer fiber. It's thought that many of the processes we're going to describe with DNA, whether it's replication or transcription, is going to happen at the level of the 30 nanometer fiber, where we relax particular regions of this 30 nanometer fiber, and then our polymerases can read them. So this is called a loop domain. So here's a 30 nanometer fiber. Here's our histone genes. What we often find in 30 nanometer fibers is within this fiber, we often have families of similar genes. For instance, we could have beta globin genes in one of these loop domains. We could have alpha globin genes in one of the loop domains. This loop domain contains the genes for the different histones. So H3, H4, H2B, H2A, H1 are found in this loop domain. And the beauty of that is we can regulate the supercoiling state of this loop domain, i.e. how easily it's transcribed, by controlling the supercoil within this loop domain, and we don't have to control what's happening in another loop domain. So this gives us the ability within gene families to regulate one region of DNA independently of what's happening within another region of DNA. And here's our nuclear scaffold, and there's proteins that are called condensins that are actually involved in the organization of these loop domains. Here's the final result of all of this. So that would be a loop domain coming off right there. And this would be a loop domain coming off right here. So this is a metaphase chromosome, which is just about as condensed as you can condense DNA. So during our cell cycle, DNA and our chromosomes exist in different states of condensation. So during G1, which is gap 1, during interphase, the DNA tends to be highly uncondensed. So during G1, we have a lot of transcription going on. We have a lot of protein synthesis going on, and our DNA is uncondensed. It's uncondensed in S phase, where we're doing our replication. In G2, starting to organize the cell to get ready for cell division. And it's only when we reach mitosis that we highly condense and we separate chromosomes. And you all remember prophase, metaphase, anaphase, telophase, etc. There is a legend that goes underneath this picture that explains what's happening at each one of these steps. So that completes L9a.